Hello everyone, and let's do the next part of the chapter, useful bacteria, and we'll also learn about the antibiotics. Now firstly we learn what are antibiotics. So antibiotics is a chemical substance that is produced by a living organism which can stop the growth of some disease producing bacteria and fungi. We have all taken antibiotics in some time of our life. Okay, so it is very important for us to know what are these antibiotics. Well, they are chemical substances which are produced by living organisms, and these living organisms, these chemical substances can stop the growth of the disease causing bacteria or the disease causing fungi. So they are mostly used for some infections. It could be an eye infection or a ear infection or chest infection. Okay, there are different types of antibiotics available in the market depending on the type of infection you are suffering from. Some of them are streptomycin, chloromycetin, there's tetracycline and penicillin, etc. Now moving on to the first antibiotic that was first discovered accidentally, it was nothing other than penicillin. Okay, now we'll study how it was discovered. So it was the first produced antibiotic and was produced from a fungus. Okay, mind you, not a bacteria but a fungus. So in 1929, it was Alexander Fleming, okay, a young biochemist, who was observing that some bacteria growing in the culture were destroyed by an accidental contamination of a mold. And the name of this mold was Penicillium chrysogenum, which is now known as Penicillium notatum. Okay. I think there's a the newer name is Penicillium chrysogenum and the older name is Penicillium notatum. Okay, so he accidentally discovered that this mold, okay, growing next to the bacteria, okay, that he was studying, it destroyed the bacteria. Okay, so the bacteria got destroyed by this culture. This mold produced a substance that suppresses the growth of many bacteria, and this substance was called as penicillin. Remember, penicillin is a product of a mold, okay, rather of a fungi and not of a bacteria. So another antibiotic was later discovered by another biochemist. He was known as Selman Waxman. He discovered again accidentally again from bacteria a new type of antibiotic. It was known as streptomycin and was discovered from the bacteria. The name of the bacteria was Streptomyces griseus. Okay, so Streptomyces griseus was used against tuberculosis, penicillin, of course, against tetanus and diphtheria, and you have chlorotetracycline that is used, that is the source of it is Streptomyces aureofaciens, and the disease it treats is a typhoid. You also have another antibiotic that is erythromycin. It source is Sacropolyspora erythracea and it is used to treat rickettsial fevers. Okay, so these the main ones are penicillin and streptomycin. Now, what are the criteria of a good antibiotic? What are qualities that a good antibiotic should have? Well, the first one, it should be able to kill a variety of disease-producing microorganisms. Okay, it cannot target just one. If it targets two or three disease-producing microorganisms, it's a very good antibiotic. This means that it should be a broad spectrum antibiotic. It should not produce undesirable side effects. Okay, you must have heard while taking antibiotics there are plenty of side effects. Okay, your body loses a number of vitamins. Okay, number of things, nutrients are lost from the body, but this should not happen ideally if you're using a good antibiotic. So it should not produce undesirable side effects. It should not kill the normal bacteria of the host. You know, there is a normal bacteria also living in your body. Okay, so that should not be killed by the antibiotic. Normally that is what happens. Okay, when you take antibiotics your body becomes deficient in vitamin B, which is the bacteria living in your gut is producing it, in your intestine, large intestine is producing this vitamin, but then it gets sort of stopped. Okay, it stops producing it, and that should not happen. Okay, it should not kill the wrong bacteria, it should kill the right bacteria. So that is the thing. Other uses of antibiotics as food preservatives. Many of these antibiotics are used as food preservatives, especially in preserving fresh meat, fish, etc., for treating the animal feed, for controlling the plant pathogens. plant pathogens means the disease causing bacteria. Okay, bacteria also affects plant growth. Okay, so it can be used for that also. Now we come to the next part, that is about serum. Okay, now what is the meaning of serum? It means the blood plasma from which the fibrinogen is removed. Okay, so the clotting part is removed, and so it is her blood plasma from which fibrogen, it does not have fibrinogen. It may contain chemical substances like antibodies and antitoxins, and that's exactly what we want. Okay, so toxins are poisonous proteins which are released by pathogenic bacteria during the growth or upon their breakdown after they die. What is an antitoxin? Just the opposite of toxin. So toxin is a chemical, it is a poisonous protein. Okay, which is released by pathogenic bacteria. Pathogenic means the one that causes diseases. Okay, so that they break down and they will act on the organism. Okay, so and they can do that even after they die. But what is an antitoxin? Just the opposite of that. It is a substance that is produced in the animal body which reacts with the poison produced by the invading germs. So it will kill the invading germs. So toxins are poisonous proteins which are released by the pathogenic bacteria, whereas antitoxin, it is produced by the animal body to counterattack the poison that is introduced into the body. Now we come later, we'll delve more into serum. Serum contains the active toxins of a particular pathogen, because it is some animal has got infected with some certain bacteria and its body has produced the antitoxins. So these antitoxins will be contained in the serum, okay, of the blood. They are used against bacterial invasion. It is produced by introducing a small dose of bacterial toxin into the blood of a healthy animal like the horse or the cow. So how do they prepare these serums? So it's normally done with with a horse. Okay, to prepare the serum done to a horse. So they introduce a small amount of toxin into the bloodstream of the horse, okay, of a healthy horse. Okay, so what happens? The horse starts creating a antitoxin, okay, to counterattack the toxin that is introduced. So its body produces a antitoxin to neutralize the effect of the toxin, and they do this repeatedly. Okay, repeated injections are given of the toxin into the blood so that the horse can produce maximum of antitoxins. Okay, now later on what they do is they remove the blood of such a horse, okay, which has been injected with the toxin, okay, and they chill it. Okay, they put in the freezer, it is freezed to a certain temperature, and what they bring out is a serum. Okay, it separate the liquid and the solid part of the blood from the serum, and now the serum, which is a straw color liquid, separates out, okay, because of the freezing that you have done, and it is known as a serum. That is known as a serum. Now, snake bite treatment also involves a similar process, injecting the particular antivenin produced from the body of horses in the same manner. So you can even treat snake bites, a person who has got a snake bite, with this type of, in that case it is known as antivenin. Okay, so in that case they must have put the poison of the snake, okay, the venom of the snake into the horse's body. Okay, so that is known as antivenin, and this is known as a serum. Now we come to the E. coli bacteria. The human genes are introduced in certain bacteria like Escherichia coli, that is also known as E. coli. These bacterial cultures, they grow very rapidly, and what is produced is a hormone insulin, which was the first substance that was produced by E. coli bacteria. So this is also another advantage of a bacteria. Okay, this type of bacteria, how it is useful to human people to produce humankind to produce the hormone insulin. Now we come to some serum compounds again used in the treatment of two main diseases, hemophilia, okay, a disease to do with the clotting of blood clotting factor 8 for the treatment of hemophilia A, and blood clotting factor 9 for the treatment of hemophilia B. So these are also serum compounds produced in a similar way. Now we come to the next part, that is known as vaccines. Okay, what is a vaccine? A preparation consists of weakened germs or dead germ substances. In this case, there is no serum. it is having a weakened germ in it. Vaccine mean, what does vaccination mean? Introduction of weakened germs or the red germ substances in the body for developing resistance to a particular disease. So once you get the vaccine, your body gets developed a resistance to that particular disease. Vaccines are available for a number of diseases, as you know: cholera, typhoid, measles, chickenpox, etc. On injecting the vaccine, what happens? A person gets a disease in a mild form. Okay, a mild form, and his body gets stimulated to produce antitoxins, and this antitoxin provides a sort of immunity against further attacks. So what happens is, if a particular vaccine is introduced into the person's body, the person will get, okay, what is actually that vaccine? It is a weakened germ only that you are introducing into that body. So that person will get that same disease in a mild form, okay, not the grave form, a very mild form. So when he gets it in a mild form, his body will again produce the antitoxin to counter the effect, right? So this antitoxin will produce a immunity, okay, against the attack of the particular disease. Okay, so this is what they are presently doing, finding the vaccine for COVID, but it's taking a bit longer because of the fact that they don't know anything so much about the COVID-19. So they're still under a lot of research, and it's changing its form. So when they're introducing the new vaccine into the person's body, it is changing. So the person is not getting the maximum benefit of it. Okay, and there are problems like that. So, but this is basically to make you understand what actually a vaccine is. Okay, it is a germ substance being introduced into the body, unlike the serum. The serum is a blood plasma, okay, that is being introduced into the body of a person who has got the disease. Hmm? Now, what is an antitoxin? And common vaccines, we'll study about antitoxin. A substance produced in the animal body which reacts with the poison produced by the invading germs. Okay, the common vaccines from bacteria are the killed bacteria for TAB vaccine for typhoid, the living weakened bacteria BCG for tuberculosis. So these are two common vaccines which are obtained from bacteria. Okay, so it's the TAB vaccine, okay, for typhoid, and the BCG vaccine for tuberculosis. So these are two main vaccines. Now we come to toxoids. What are toxoids? Inactivated toxins. So if a person's individual's body has got the toxins, okay, some of these toxins they like remain dormant, okay, so they don't get activated, okay, which are still, they are unable to stimulate themselves, okay, so these remain dormant in the body. Okay, so such still toxins of the particular bacteria which can still stimulate the production of the respective antibodies, such as those producing immunity against diphtheria and tetanus. So what happens? These toxins can again help. Okay, they can help to counterattack a disease that has come into the body of the same person. Okay, so toxins are not produced by bacteria, but they can be used as vaccines. Okay, so what are toxins? They are inactivated toxins, okay, of a particular bacteria which can stimulate the antibodies. Okay, so they are living, they are remain there, but they can produce antibodies whenever required. Okay, so you can have these against diphtheria and tetanus. Now we come to the role of bacteria in agriculture. You have already studied in your lower classes about this bacterium known as rhizobium. Okay, now what is rhizobium? It is a bacteria that lives in the root nodules of leguminous plants. Okay, just like beans. Okay, what are leguminous plants? They are plants which are rich in proteins. Okay, now these plants rich in proteins, in their roots you will find like these nodules in them. Okay, they are small outgrowths. These bacteria, what they do is they live over here. Okay, so this is a symbiotic relationship where the bacteria is taking shelter from this plant, that is a leguminous plant, and the leguminous plant in turn is getting the benefit of the rhizobium. The rhizobium, what it does do, it fixes the atmospheric nitrogen, in the soil in the form of nitrates. It is able to convert the atmospheric nitrogen in the form of nitrates. So rhizobium bacteria live in the root nodules of leguminous plants such as beans. These bacteria pick up the free nitrogen from the soil and convert it into soluble nitrates and gives it to the plant. These nitrates are used by the host plant. Okay, these host plant, they need to produce a lot of proteins. So it can produce a lot of proteins from the nitrates. Okay, as I told you earlier, nitrogen helps in production of proteins. Azotobacter and Clostridium bacteria convert it into ammonia, okay, with hydrogen, which is converted into ammonia acids and nitrates, enriching the soil. Okay, so these other bacteria, they convert this nitrates again back into ammonia, okay, so which can be converted back into nitrates, and this can enrich the soil. So that's the role of bacteria in agriculture. Now we also know about the nitrifying bacteria. What are nitrifying bacteria? They are able to convert the nitrites and nitrates, okay, nitrites and into nitrates, and these nitrates can then be taken up easily by the plants. Okay, so understand this is known as a nitrogen cycle. Okay, here you have the nitrogen cycle. Here you have the atmospheric nitrogen. Okay, this atmospheric nitrogen, it gets fixed into the soil, as I told you, by the rhizobium bacteria or any other bacteria that fixes the nitrogen to the soil in the form of, and there can be even acetobacter, which is converting it into ammonia and keeping it into the soil. Similarly, industries that give out nitrogen, okay, that is also converted into ammonia. Okay, then you have electrical fixation. This takes place during thunder, lightning. Okay, during lightning, there's prospering nitrogen is getting converted to nitrates and enters into the soil. Okay, now what happens is, again over here you have this ammonia. This ammonia, it gets converted into ammonium compounds. How does that happen? First, it gets converted into nitrate, okay, sorry, nitrite by a bacteria known as nitrosomonas. Okay, so it is getting converted into nitrites by nitrosomonas. This nitrites is getting converted to nitrates by nitrobacter, that is another bacteria. Okay, so this is happening here. Nitrosomonas getting converting the ammonia into nitrites, and nitrobacter converting it back into nitrates. Now this nitrates is easily taken in by the plants. Okay, so these uptake is by the plant biomass, okay, or the plant families. Now these plants are eaten by who? The animals. So these animals consume, that way they are indirectly consuming the nitrates, or even our bodies. Okay, we also take in the plant, okay, which are having the ammonia converted into nitrates. Now, when these plants and animals die, what do they become? They become the decaying biomass. Okay, so their bodies get converted again back into ammonia, okay, by the process of decomposition. Okay, and again this process that continues. Okay, so this is like a cyclic process, so it is known as a nitrogen cycle. Now, again, what is happening after this? There are certain bacteria in the soil like pseudomonas, which what they do is they convert this nitrates back into atmospheric nitrogen. Okay, so nitrogen gas is released back into the atmosphere, and then this continues again. Okay, like this, it goes on, and then denitrification again back to the atmosphere, again gets fixed, again gets converted into nitrites, nitrates, then taken in by the plants. They are decaying, gets converted to ammonia and back, and then this denitrification. Okay, so this is a cyclic process, and hence it is known as a nitrogen cycle. Now, what is the role of bacteria in industry? We know that bacteria is also used in various industries for positive reasons, obviously, for tea curing, bringing different flavors in the tea. Okay, so, and this is a process of actually dehydrating the tea. Okay, dehydrating the tea and converting its flavor into a very tangible one which people will enjoy drinking. The second one, it is used in leather tanning. Okay, so leather tanning is brought about by sunlight when certain bacteria can break down the soft perishable parts of the skin. So what happens is there are certain parts of the skin of the animals, okay, from which you're making the leather, which can destroy the leather later on. okay, certain parts. Okay, so it is good to treat it with this bacteria. So this bacteria will break down the soft parts of the skin. Okay, so it will not harm the leather product. Next, you have bacterial spoilage of food. Now we're going to learn all the negative impacts of bacteria. Botulism is very serious food poisoning, especially found in tin foods and cans. Well, the metal cans in these food is stored gets distended, okay, with their ends. At their ends with gas which gushes out when it is opened. Such suspected cans should be discarded immediately. Therefore, preservation of food and making disease free is very necessary. Certain times what happens if you keep the tinned can, okay, beyond its expiry date, you will find that the can gets little distended. Okay, so this is because the bacteria is developed in that, okay, and it is releasing a gas. Okay, so when you open it, this comes out. Okay, so these cans, which when you open it, you get the expelling of gas. Okay, you should just discard those tins, those cans. You should not use them because that can lead to botulism, which is a serious food poisoning issue caused by bacteria. Now we come to how to preserve food, how to keep food free from bacteria. That should be our aim if you are having an industry of jams, pickles, or something like that. The first method is sterilization. What is sterilization? Boiling water to 110 degrees Celsius at an increased pressure that will kill even the spores. Even the spores will die at 110 degrees Celsius. So during canning of food, it is heated to kill the bacteria present in the food. Second method is salting. But if you mix salt with these products that you want to preserve, salt is like a preservative. Okay, it will preserve the food. It will prevent the degradation of food under normal conditions. So if you see, if you want to retain pickles, pickles have a large amount of salt content in them. The reason for it is because it has to last for more than a year. That is why you're putting so much of salt. Also the dried fish and also cheese also, it contains a lot of salt. This treatment, what is it called? It's called as curing. Okay, putting salt and keeping it for a long duration of time is known as curing. Now what is dehydration? Dehydration means you are dehydrating the substance, you are removing the moisture from the substance. So that is done, to prepare pappers, to prepare candies like avla candies, imli candies, okay, grains, to keep grains for longer duration, fish, okay, drying the fish in the hot sun, drying meat, vegetables also dried, the bitter gourd, okay, that is also dried and you can fry it and eat it. Okay, so these are all methods of dehydration. Now we come to irradiation. As the word suggests, radiation means you are going to pass radioactivity, UV radiations through it. Okay, so UV radiation is used to sterilize even the air in the schools, in hospitals, okay, to kill the viruses. Also can be, certain viruses can be killed. I know about coronavirus. It is also used to kill the spores and molds in bakeries also. Then you come to refrigeration. Microbes do not, why do we keep foodstuffs in the fridge? Okay, so we don't want bacteria to grow on them, because we know at a degree, at a lower degree, between 4 to 0 degrees Celsius in the refrigerator, the bacteria will not be able to survive. So the food is free from bacteria if kept in the fridge. So microbes do not grow and multiply or below the freezing point of water, and hence vegetables and fruits can be stored by chilling them and keeping them for fresh for longer duration. The domestic fridge, it will cool at 1 to 5 degrees Celsius, and if you keep in the deep freezer, it cools between minus 20 to minus 30 degrees for long time storage. If you want to keep your certain items for a longer period of time, you need to keep it in the deep freezer, okay, where it is minus 20 to minus 30 degree centigrade. Now, a third, last method is pasteurization. So this is done in case of milk. It is a technique of partial sterilization, usually applied to milk. Milk is heated to 60 degrees Celsius, and how much time? It is boiled for 30 minutes at only 60 degrees. It is taken care not to exceed 60 degrees. Okay, and then it is chilled quickly. Now, what is this method of chilling quickly? This is known as pasteurization, so as to kill certain bacteria. but you don't want to kill certain bacteria, otherwise you'll not be able to use the milk in the proper way. You will not be able to produce the curd from it. So that is why you don't have to totally degrade it. Okay, you need to only heat it to 60 degrees Celsius. It does not completely sterilize the milk. Souring bacteria may be present. Okay, so that is why we need to keep certain bacteria by keeping the milk cool, but they do not multiply. Okay, so you can afterwards, that's why they are doing that heating suddenly, then cooling suddenly, so that only certain bacteria die, certain bacteria are retained. Okay, to promote the, so that they do not multiply. Okay, you can keep it in the fridge later on. Strong concentration and chemicals. Now when we make jams, when we make pickles, okay, they do not get spoiled. Okay, because when we make jams, we are putting a lot of sugar in it. When we are making pickle, we are putting salt in it. So what are we doing? We are increasing the concentration of certain things in it. Okay, increased concentration of solutes causes plasmolysis, death of the bacteria or the mold. So when we are putting the solute, a solute in this case is the sugar and the salt. So it leads to a process known as plasmolysis death. Okay, so there's a death, plasmolysis death of the bacteria at such a high concentration of solute. Sometimes the chemical sodium benzoate is also used in the making of especially squashes and in making of sauces. Okay, in tomato ketchup and all, this sodium benzoate is added to the tomato puree. Okay, so as to kill the bacteria. Now, just as human beings and animals suffer from bacterial diseases, even plants suffer from some bacterial diseases. For example, there is a black rot of the mustard, the black rot of the cauliflower, you can see here, and here you have the bacterial blight of the cowpea. Okay, so these are all examples of the bacterial diseases caused in plants. Also in animals, the anthrax, that is swelling of the body of the cow and reduced milk yield. Bovine tuberculosis, that is lungs get affected and they have a dry husky cough. Okay, so this is comes later on in the life of the animal. Okay, so but these are caused by bacteria. The resistance of the animal becomes less, they get this bacterial infection. Now, human beings also suffer from bacterial infection like whooping cough, cholera, tuberculosis, diphtheria, typhoid, tetanus, and luckily we have vaccines for all of them. Okay. Now, what are bioweapons? A little note on bioweapons. Germ bombs are produced by some countries in order to fight with the opponent country, and these bombs, they can explode and they can cause disease, producing germs everywhere. These germs may make an epidemic shape, or now like you can see the pandemic shape of the coronavirus. It's like a bioweapon in order to destroy other countries. Okay, and it can widespread death generation after generation. As you know, I don't have to tell you much. Okay, as you are aware of it. Right now we are suffering because of the virus, okay, which is a sort of a bioweapon. Thank you everyone for watching the video. Goodbye.